Hi, students, and welcome to the Sound of Science. Today, we're going to be talking about gene expression and mutation and the impact that mutation has on how our genes become expressed. Okay, so the first thing you need to know is that some genes are going to be expressed in some cells and others are not. And it can change depending on which cell we're talking about. And this deals with cell specialization. So your cells all have the same DNA in it, but sometimes only certain portions of that DNA gets used. And we see this a lot as cells start to go through development, as they start to specialize, and certain areas of the genes really get turned on and used, and then later on they might be repressed or they stop being used. And this means that your skin cells are only using the genes that skin cells need, while your kidney cells are going to use the genes that kidney cells need. And there's really no reason for your skin cells to act like a kidney cell. So this is actually a really brilliant system that our body utilizes. If the gene is being promoted, then it's transcribed and it happens. If it's being repressed, it stops transcription. It keeps it from being transcribed into RNA. So that gene never gets used to make a protein. Now, like I said, it's different for each cell. So certain types of cells are going to have certain genes that are promoted. Certain types of cells are going to have certain genes that are repressed. We see this type of stuff. It's very, very easy to understand when we see it in things like bacteria. We can actually see the promoters and the repressors. When we move on to eukaryotes, it gets a lot more complicated. And there are transcription factors that allow the polymerase, RNA polymerase, to attach, or they prevent it from transcribing a section of DNA. It just sort of depends. It's a little bit more complicated, but really, as long as you understand that there are factors that prevent transcription or factors that allow transcription, then you kind of got the idea of how this is going to work. Okay, cell specialization is really important, and this happens throughout development. So as a fetus starts to develop and it starts to get these specialized cells, those cells have certain portions where some DNA needs to be used, but later in development, that DNA doesn't need to be used. And that is how cell specialization works with us multicellular organisms. So the genes that determine when things develop in an organism are actually called homeobox genes. And that's this word right up here, homeobox genes. Homeobox genes help to determine the organs that develop in each body part. So obviously, when I'm trying to develop arms, I don't want to develop a heart in the middle of that. That's not the right place for the heart to go. And so these homeobox genes just kind of keep things on track. Like, okay, right now we're developing the arm buds and the leg buds. So those are the genes we need to focus on. A mutation in the homeobox genes can actually cause pretty serious malformations or mutations. So if you look down here, these are flies. This is a perfectly normal fly. This fly hatched out with its antenna in the correct location, right? This fly had its homeobox gene mutated prior to development or during development. And so the part where it is supposed to say grow antenna, instead now it's like, oh, nope, now it's time to grow legs. And so instead of antenna, this little fly ended up with legs where the antenna would have been. Those types of mutations can be very detrimental. If you look over here and you see this little boy, this is actually what we consider a thalidomide baby. Thalidomide was a medication that was developed. It was actually given to pregnant women without testing on what it does to fetuses. And what they discovered is that thalidomide causes mutations in these homeobox genes. And so babies that were born to mothers that had taken thalidomide during the first trimester were often born without limbs. Sometimes they would have no lower half of their body. So that's just an example of what homeobox genes can do. Okay, epigenetics. So I always like to tell people that epigenetics is why YOLO doesn't work. So epigenetics just means above genetics. there's these methyl groups, and this is another chemical, another compound, and it actually stops gene transcription. Then you have some other groups that are histones with acetyl groups. Again, more chemistry, that can actually open up the chromosomes, which increases the amount of transcription that can happen in specific genes. So methyl groups and these histones with acetyl groups get added based on your exposures to things. It could be your exposures to chemicals. It can be exposures to foods. What's in your diet? What kind of exercise are you doing? All of those things actually influence how your DNA functions. So it's going to work for you, but not only that, it actually is going to affect your offspring. You can pass on those methyl groups to your offspring, which means that your DNA is being affected by your ancestors, your great-grandparents or great-great-grandparents, what they ate, where they lived, what they were exposed to, what viruses they were exposed to. All of those things influence how your DNA does its job. So that's why YOLO really isn't YOLO, because what you do now affects those that are going to come after you. Let's talk types of mutations that you might see. So point mutations come in really two different forms. Three if you take them each specifically, but you have a substitution mutation. Just a single base pair is replaced with a different base pair. So let's say that it's supposed to be CAT, and now instead it's going to be GAT. It's a single base pair change. But as you saw with the codon charts, a single base pair change can actually have a pretty huge impact. Now, frame shift mutations are also point mutations. That means that a single base pair is changed or a couple of base pairs are changed. But instead of taking one out and putting one in, like with a substitution, now you're either going to be taking one out in a deletion or putting one in as an insertion. And what this does is causes a frame shift. So if you look at this first line of codons here, we have cat, tac, gag, right? These are actually all words, right? But if we add a G between this C and this A right here, it actually causes the entire codon to shift. So now instead of CAT, our first one is CGA, which is a completely different amino acid. TAC goes to TTA, and GAG becomes CGA, with an extra G kind of hanging out in the end. This insertion caused what we call a frame shift. Now, instead of the ribosome reading these codons, it's actually going to read the wrong codons. It's making codons that weren't there before. And so that frame shift mutation can actually cause very, very, very severe genetic disorders because it literally changes every amino acid that comes after, or nearly every amino acid that comes after. So the protein won't be the right shape. That's the problem with any mutation. If it causes the protein to have the wrong shape, then we know the protein's not going to work well. And these are the individuals that end up with some type of genetic disorder. Chromosomal mutations, on the other hand, involve larger portions of the DNA. So a large portion of the chromosome is not included. It might be duplicated. Sometimes it's inverted, like literally you can take a portion of the chromosome and it actually gets flipped upside down or flipped over. It can be translocated. It can be picked up and moved on to another chromosome. Out of all these mutations, these are some of the examples of the genetic disorders that we start to see. So, sickle cell anemia is a substitution mutation. For those of you that don't know, sickle cell anemia is where your red blood cells, instead of being nice fat round cells, actually get sickle shaped. And those sickles end up attaching to each other and they get kind of hooked and they get stuck in the little capillaries that basically run through your body. And so individuals with sickle cell anemia can end up with a sickling crisis. That means that they are in severe pain, particularly in their joints where these little capillaries are. They really struggle with moving oxygen throughout their body because red blood cells are designed to move oxygen around. So all of those things can happen with sickle cell anemia. Sickle cell anemia actually does have a benefit if you live in a place where malaria is prevalent. The malaria parasite that you get can't infect red blood cells that are sickled. So individuals that have some sickling might actually receive some protection from malaria. Huntington's disease is an insertion mutation. This is causing a frame shift. Individuals without Huntington's, this is what their brain's going to look like. Individuals with Huntington's, they actually start to have sections of their brain atrophy. So you can see that portions of the brain are just disappearing. Tay-Sachs disease is another frame shift, and it's caused by a deletion. This time, instead of adding one, like with Huntington's, we actually remove one. This is another one that affects your nervous system, and it literally allows lipids, fats in the brain, to start eating away at nerve tissue in the brain, these nerve cells. The end result is, again, a fatal disorder. Okay, so Down syndrome. This is a chromosomal mutation. That means there's a change to the chromosome. So this right here, this whole picture, is a karyotype. Karyotypes are as if somebody went into your cell, into your nucleus when it was getting ready to divide, took out all of your chromosomes and then lined them up based on which chromosome they were. They lined them up in pairs. So here we actually have our first one. It's going to be our largest chromosome, and they match banding. So the banding of the chromosome matches its homologous chromosome. That just indicates where the genes are. So with Down syndrome, when you look at a karyotype, you see that we have 22 pairs of chromosomes. And then our sex chromosomes is our 23rd pair, that's X and Y. So there's 46 chromosomes total. Except this individual has 47 chromosomes because they have an extra copy of chromosome 21. That is what causes Down syndrome. Just having an extra copy of a gene causes all of Down syndrome's symptoms. We actually have two karyotypes here, and I want you to use the karyotype to determine which one of these is a boy and which is a girl. And I'll give you a hint. You're going to look at this 23rd pair, these sex chromosomes. So the sex chromosomes here, we have an X and Y, and here is two X chromosomes. So which one's the boy? XY is the boy. XX is the girl. So that's another thing that we can determine from karyotypes. Now, if we look at this karyotype, you can see that they have normal chromosomes until we hit 13. 13 has an extra chromosome. This is actually called trisomy, which means 3 chromosomes. Trisomy 13. This individual also happens to be a girl. They have 2 X chromosomes. Trisomy 13 is another disorder that we definitely, it causes a pretty severe set of symptoms. And again, caused by just an extra chromosome. So what about this individual? Well, if we look through, all of these chromosomes look pretty normal. I don't see any extras. And then I get here and like, oh, 2 X's, they're a girl. Oh, they have a Y chromosome. So they're also a boy. This is another type of genetic mutation that's actually pretty common. This individual has 2 X chromosomes and a Y. They are actually going to be biologically male, but because of this extra X chromosome, they are likely to be sterile. They may have more difficulty with obesity. Remember when we did meiosis and we talked about calyx, and this was calyx's mutation. So we actually see this in humans as well. Are all mutations bad? Some mutations are going to be bad for the individual that gets the mutation. Other mutations are actually helpful, or they don't have any effect at all. If it's a substitution mutation, it actually may end up creating the same amino acid or coding for the same amino acid that the original did. So that would actually have no impact whatsoever. Or it could be such a slight change that it actually doesn't affect the overall shape of the protein. The protein's still able to work or works nearly as well. In plants, instead of being 2N, sometimes we can actually force them to be polyploidy, where they have 3 copies of all their chromosomes or 4 copies of all their chromosomes, unlike us that have 2 copies. With us, if we have 3 or 4 copies of our chromosomes, we're going to have some serious genetic disorders. With plants, it actually just makes them stronger and larger. So it actually works out really well for them. For bacteria, one of the mutations that they develop often is antibiotic resistance. Very bad for us, very good for the bacteria. It allows it to avoid being killed by antibiotics. So some mutations, like I said, can be helpful. A vast majority of mutations actually occur in places that aren't even coding sections of your DNA, and they don't have any impact whatsoever.